Dear class 9 children, we will continue chapter number 9, chapter number 7, that is respiration in plants. In the last session, we discussed about the definition of respiration and also some other related terms. So today in this session, first we'll talk about types, okay? Types of respiration. So there are two types of respiration. They are aerobic respiration and anaerobic respiration. Okay, aerobic and anaerobic respiration. So aerobic respiration is a type of respiration which takes place in presence of free oxygen. So this respiration can also be represented by the chemical equation which I am going to write here. See, C6H12O6 plus 6 molecules of oxygen in presence of enzyme, okay, produces 6 molecules of carbon dioxide plus 6 molecules of water plus 38 ATP. So this is the complete chemical equation of aerobic respiration, where you can see the role being played by oxygen here. Okay, where oxygen is very much important. Okay, in presence of free oxygen only, the process can be, can be continued. Okay, if it is not there, then the process will stop. See here. So what happens here is that you see 1 mole of glucose. Okay, 1 mole of glucose, C6H12O6, that is glucose. So 1 mole of glucose is oxidized by 6 molecules of oxygen in presence of enzyme to produce 6 molecules of carbon dioxide, 6 molecules of water, and large amount of energy, that is 38 ATP. So ATP means adenosine triphosphate, which is an energy rich compound. Okay, this is the chemical equation of aerobic respiration. Okay, whereas the other type of respiration, that is anaerobic respiration, takes place in absence of free oxygen, where oxygen is not needed. So that can also be represented by the chemical equation which I am going to write here. C6H12O6 directly gives to C2H5OH, C2H5OH, that is ethanol. Alright, then 2 molecules of carbon dioxide plus 2 ATP. See here, now you can compare these two equations. Okay, you can see here. So here no oxygen is needed. Directly C6H12O6 is giving 2 molecules of ethanol plus 2 molecules of carbon dioxide in very less amount of energy, that is 2 ATP. That means 2 molecules of adenosine triphosphate. Okay, see here no need of oxygen. You can see here. Okay, so if you know these two equations of aerobic and anaerobic, then you can talk a lot about these two types of respiration. You can even compare them. Okay, you can say a lot about the differences between aerobic and anaerobic respirations. Okay, so this aerobic respiration takes place in all the higher plants and animals, all right, and also in unicellular organisms, excepting a few. Okay, whereas this anaerobic respiration takes place in some unicellular organisms like bacteria. Okay, in some bacteria, and also in fungi. Okay, the process take place. Okay, fungi, and in some bacteria this process continues. Sometimes this in our case also sometimes. Okay, the what happens is that during vigorous exercises, okay, when we are working hard, okay, when you are playing the football for the first time in the season, so that time what happens is that due to the shortage of oxygen there in the body, the skeletal cells, okay, the skeletal cells respire anaerobically, respire in absence of oxygen. Consequently, what happens there is that you see there in the skeletal cells, the accumulation of lactic acid takes place. Okay, accumulation of lactic acid takes place, which which gives us pain. Okay, because of which we feel pain there in the muscles, there in the skeletal cells and all. That's all because of the formation or accumulation of lactic acid there in the skeletal cells due to anaerobic respiration. Okay, but when we take rest for some days, okay, after some days what happens that this lactic acid, the accumulation of lactic acid will be channeled back to, or the incomplete breakdown of that glucose will be channeled back to the aerobic respiration. Then the problem will be solved. Then all those lactic acid will be vanished, will be will be removed from the body, and no pain will be there. Then we get relaxed. Okay, this is how sometimes we do. We also respire anaerobically. Okay, not throughout the body, but sometimes they're in the skeletal cells during the vigorous exercise. Okay, some other point that I want to tell you is the point of difference between these two, that here in aerobic respiration complete breakdown of glucose takes place. That means glucose will be completely broken down, whereas here the glucose, you see the glucose being incompletely broken down. So here the huge amount of energy you see being produced here, 38 ATP, whereas here only 2 ATP. Okay, so these are the some points of differences between aerobic respiration and anaerobic respiration. Okay, okay, now we'll see the experiments on respiration. Okay, so first experiment we have got out here is to prove that oxygen is used up in respiration. As you all know that for respiration oxygen is needed. That means oxygen is used up by the living organisms for respiration. Okay, to prove that, here is an experiment. All right, so here first what you do is that this is how you'll prepare an apparatus where you see the flask A and flask B, as you see here shown in this setup. In flask A, what you see is that you see the things arranged like that. Okay, so before arranging all those things there, first what you do is that you need to have the flask A and flask B here. Flask A is here, and you on the side you see the flask B. So then what you do is that you need to collect some bean seeds or pea seeds. Okay, some means you see like 60 such bean seeds or pea seeds you have collected. Okay, then divide them into groups. Suppose if it is 60, then you make it 30 each. Okay, so 30 bean seeds, first you need to soak them in the water for about 20 parts. Okay, then keep them their own wet cotton wool. Okay, and put them all there in flask A. Okay, whereas the remaining 30 seeds need to be killed by boiling them. Okay, you have to boil them so that they are killed. Okay, then they are to be treated with some antiseptic chemicals like carbolic acid. Okay, so that bacterial activities are kept away. Otherwise, what happens if they are not treated with such chemicals, then bacterial activities can occur there, and those bacteria present out here would respire and use of oxygen present there. Okay, so that they are to be treated with such chemicals so that no any bacterial activities are there. Okay, then those dead ones need to be kept here on on a cotton wool and to be kept there in flask B like this. Okay, then what you do is that in flask A and B, in both the flasks, what you see here is that you see the test tube hung. See the test tubes you see being hung both sides. This test tube contains soda lime. Okay, soda lime, which is a mixture of sodium hydroxide and calcium oxide. Okay, and its function is to absorb CO2. If there is any carbon dioxide, okay, will be absorbed by soda, soda lime present or contained in this test tube CM. Both sides soda lime is there, and its function is to absorb carbon dioxide. And this is hung in such a way that its free end of the thread is taken out through this cork. Okay, so that they can be nicely hung inside. Okay, now what you do is that you see the delivery tube also coming from both the flags. Flags A, you see the delivery tube coming out and going into the beaker down there kept on the table. And this beaker you see containing water, and other side also, and from the other flags also you see the delivery tube coming out and going into that beaker which has already contained the water here. Okay, now what you do, you just arrange them this way. Okay, they are just kept this way in this stand. Okay, they are arranged, they are all supported by the stand in this way, as you see there in the figure. Okay, now leave them for a few days. Okay, and after a few days you observe the changes here. What changes you can see here is that you see here in flax A, you see all those bean seeds germinating. Okay, you see all the bean seeds germinating very well. Okay, you can see the bean seeds germinating. And in the other side, in the flax B, you see the seeds not germinating. Okay, why? Because they are all killed. Okay, they are all killed. That's why. Okay, here you see the seeds germinating. Okay, in the meantime, you see the water level here. Water level here rising. Okay, rising in this delivery tube coming out of this flax A. Okay, the volume of oxygen used up here can be seen with the, can be calculated by the rise of, water here in this delivery tube coming out of this test tube, flax A. Whereas in the other case, okay, the delivery tube coming out of this flax B will not show any rise in the level of the water. Okay, so from this what we understand is that here something has happened. What is something here? Something means the oxygen has been used up by this germinating seeds here inside. Okay, to prove that now, okay, so we are saying that there is the use of oxygen. So how do you know then? It should be proven experimentally. So to prove that, what you now can do is that you take this test tube out very securely. Take this test tube out very securely. Then what you do? Remove the cork. Then you insert a burning paper inside it. Okay, put a burning paper inside the flax B. Flax A, sorry, inside the flax A. So as soon as the burning paper is put inside, the burning paper will go off. Okay, it will not burn. Okay, it will be put off. Okay, it will be put up. It will stop, burning. Why? Because there is no oxygen. And as you all know that oxygen is a supporter of combustion. If there is no oxygen, then nothing will burn. So for burning also, what is needed? Oxygen is needed. That's why when we had put the burning paper inside, it is not burning, it stops. Why? Because there is no oxygen. Okay, but in the other case, when you take it out, when you take this flax B out and put the burning paper here inside again, what you find is that the burning paper you find continuing to burn for some time. That means that burning paper will continue to burn for some time herein when we put it there in flax B. What it supports, or what it proves that it stopped burning. Why? Because there was no oxygen, and it continues to burn for some time because there was oxygen. So from this what we conclude is that here was not oxygen. Why? Because all the oxygen present here have been used up by the this germinating seeds here, whereas for respiration, whereas here the oxygen was still there. Why? Because the oxygen was not used up by this dead seeds. Okay, so from this experiment we understand that oxygen is used up by any plants and animals for respiration. So here in this case we find the use of oxygen for respiration in the germinating seeds. Okay, now we'll see the experiment number 2. It is to prove that carbon dioxide is released or produced during respiration. Okay, as you all know that after respiration or during respiration we find the release of carbon dioxide. Okay, it is to prove, the experiment which I am going to discuss is to see that carbon dioxide is produced during respiration. Okay, so for that what you do is that uh, collect the flags A and B again. Okay, flags A and flags B. Then what you do is that you need to collect such bean seeds. Let us say again 100 bean seeds or pea seeds or whatever it is. Then what you do is that you can separate it into two halves or into two groups. Okay, into equal halves, that is 50 in one set and 50 in other set. Okay, so 50 bean seeds you need to soak them, just keep them there in water overnight, and next 50, okay, 50 bean seeds you need to boil them so that they are killed. Okay, and after killing them they need to be treated again with the antiseptic like carbolic acid. Okay, so that no any bacterial activities take place. Okay, otherwise again the same thing will be there, like the bacteria will respire using oxygen and release carbon dioxide. So to see the release of carbon dioxide during respiration, that objective, that aim of the experiment will be completely wrong again here. That will again be futile. So in order to check that one, we have again used the antiseptic to treat the dead seeds out here. Then again leave the apparatus or leave the setup A and B there for some days, and after some days what you do is that you check them. Okay, you see them. So first of all, the changes you can see out here is that those bean seeds in A you see normally germinating, whereas in B they are not germinating because no no germination out here, because all the seeds kept out here are dead. Okay, now you need to check whether the gas has been produced or not. Which gas? Carbon dioxide has been produced or not. Now see, so for that what you do, just take this out. Okay, just consider flax A first. Then what you do, just remove this cork very carefully. Okay, cork needs to be removed very carefully. Then then what you do, you'll have to tilt it down like this. Okay, you'll have to tilt it like this into the test tube. Okay, into the test tube containing lime water. Here is lime water. Okay, here is lime water. So this test tube or flax A need to be put into it by tilting it. So once you tilt it, since carbon dioxide is heavier than air, so this carbon dioxide will be coming down here and will be going into this test tube containing lime water. Then that carbon dioxide and lime water will react. So once the expected gas carbon dioxide comes in contact with lime water, it will turn to milky white. It will turn to what? Milky white. Proving that the gas released here was carbon dioxide. Okay, so that means these germinating seeds have taken part there in the respiration and have released CO2. Have released what? CO2. That's why when this CO2 comes in contact with lime water, turns to milky white. That means your lime water now turns to milky white. This is how we test carbon dioxide. So, but whereas in flax B, no any germination in there, germination you see there. Okay, so though it is very obvious that there is no germination, even to confirm it, what you can do is that you can remove it and again, pour it into the test tube containing lime water. So once you tilt it and pour it into it, you'll not see any change out here. Okay, your lime water will not turn to milky white. It remains as it is because there is no carbon dioxide. But here in this case, you see the carbon dioxide, okay, which is confirmed by you know seeing the change in the color of this lime water into milky white, which all happened when we tilted it into this test tube containing lime water. So this is how we prove that carbon dioxide is released during respiration. Okay, so today we'll do only this much and we'll continue in the next class. Okay, thank you very much.